All right. How you guys doing? Making it? Almost halfway through the grading the exams. Not done yet, but should be on track for having them back to you next week sometime. So working on that. Today we're gonna pick back up into wastewater treatment and talk about kind of the secondary treatment process, get into a bit more about what we're actually doing with the bacteria and how do we manage them. A quick reminder, I did post a Moodle quiz or two. I think I put it sometime last week, but they were available up until I think the end of this week, and then one's end of next week. So do pay attention and take those quizzes. I think one's going to be on the Flint water crisis seminar, and I might have preemptively added one for this kind of stuff. I'll double check and probably add another one or two, maybe one more before the break and then one after, something like that. Okay, and I'm not feeling rushed on giving you a homework. There'll be one last homework that'll be due kind of before the final exam, basically. So that's kind of the logistics, and so we'll get right back to the wastewater treatment here. So last time we looked at a kind of a big picture. We had the primary treatment, take out kind of the solid junk, clutter. And then we have a secondary treatment, which is primarily the biological activity. That's what we're going to focus in on. And the way we can conceive of this secondary treatment, the biology part of it, is we have our bioreactor producing lots of cells. Then we're going to collect those cells in a clarifier. We'll call this the secondary clarifier since we're in the secondary treatment step. Then we're going to recycle some of those bacteria back into the tank. So if we draw a control boundary around this whole system, what we'll notice is we have, whenever we do any sort of mass balance, we're going to be crossing that control boundary in 3 spots. We've got an input, an output, and a second output. So way back at the beginning of class, we did look at some systems where we could have multiple inputs or outputs. For the most part, I haven't really challenged you with multiple inputs and outputs type of problems, but you can imagine it shouldn't be too different, right? We could have, let's say, a steady state system that was QC not coming in minus Q1 C going out minus Q2 C going out, right? That would be, maybe plus some reaction term. We could draw it up that way. We could draw it up with multiple inputs, maybe with different inputs. We can add to this mass balance according to the input and output rates. In this case, we're going to be paying a little bit closer attention in particular to the amount of bacteria. So as we'll see in a bit, X is going to be a concentration of bacteria. So X, and we're going to grow so much bacteria that we're actually going to be able to weigh them. So we're going to be able to scoop out a mass of bacteria, filter them, separate from the water, and actually weigh that. So when we talk about the system here, we're going to be dealing with a hefty amount of bacteria growing in here. And then we can essentially track how much is leaving or how much we can control that. But the key part here is normally, if our clarifier is working properly, we shouldn't have much mass escaping. So if our clarifier is working well, there's going to be a few bacteria suspended in solution, but you're not going to be able to really weigh those as a meaningful amount. So the meaningful amount is going to be separated. This XE goes to 0. We'll look at that more in the future, but I'm just kind of pointing out the operation here. We have a separation. It's almost like a membrane. In fact, we can use membrane bioreactors that instead of a clarifier, you could use a membrane. The point is to keep the bacteria in the tank or in the system and have control over them in that manner. All right, so given that, the big picture here is we've got water coming in, water going out. There's a little bit of water, so QW here generally is small, because this is a flow rate of waste sludge that we're just going to go send to dewater and then to the landfill. So we don't actually want a lot of volume there, a high flow rate. It's going to be a small amount of water, a large amount of solids. So we'll just say lots of solids there. And in the flow rate going out, this is most of the water going out of the clarifier. So we'll say QE is maybe almost Q in, we'll call it, if Q in was coming in for the first place. So it's just a little bit less than. So most of our water is just going straight through the clarifier and going out. The difference here is we're controlling the sludge. So we have all this sludge growing, activated, so to speak. Then we have this return activated sludge line, RAS. That's going to be the return or recycled activated sludge. One thing to note here, if I could spell sludge right, we have a split in the pipe here between what's wasted and what's recycled. And if we divert a pipe into two directions, that's not going to change any of the concentration, right? That's going to be exactly the same as we had it. So whatever concentration here, so X waste, is going to be equal to XR. Again, we'll come back to this as we derive some equations, but I just want to point out the fact that if we have XW here, that's equal to the XR that we see there and that we see here, right? Because the splitting of the pipe is not going to change any of that, right? That's just some amount goes this way, some amount goes that way. The difference when we have a clarifier, for example, that does change the concentration of the solids, right? So that's going to make a difference. Okay, so that's kind of the system. One thing you'll notice with a control boundary outside of the recycle, we can ultimately do mass balance type analyses without worrying about what's happening with the recycle. So it ends up being very convenient that we can draw a control boundary way out here, right? That's where we cross. So all of that recycle stuff does not directly affect it, and we can do some simplification for our math. All right, so then ultimately the question is, how do we manage X, the amount of bacteria there? And the other point is, well, what's the actual stuff we're treating? That is S. X is the bacteria. We use those as a source. S is the food, the substrate, the stuff we're trying to treat. That's the actual waste. So we'll call that S for substrate. These are the waste materials, right? We could also call it bacteria food. It's the substrate on which the bacteria are growing. They're using this for energy, for production of cells. This is their nutrition. This is their substrate. So in the tank, what we're trying to accomplish is a reduction in S, right? And in the meantime, we need to have a production of X. So we'll produce X to consume S. That's kind of the overall system. So that's just simply saying we're using bacteria, we're growing bacteria in order to munch on the waste stuff. Again, converting these dissolved things, hard to deal with things, who knows what in there, and converting that all to solid particulate matter is very, very helpful. Okay, so let's get into this terminology. I mentioned a lot of this already, but here it is written for you. Waste activated sludge, WAS. This is that slurry of microbe soup, right? It's gross. You don't want to fall in there. Say more about that in a minute. It's constantly aerated in these systems. If it's activated, that's activating it with air, essentially. And by bubbling air through the system, we are also giving it some mixing conditions. So effectively, we're going to be using these activated sludge basins as if they were CSTRs. Okay? In practice, maybe sometimes they're a bit long, you know, if it's a chamber like this. In practice, we don't always get mixing from here over to here, but there's enough mixing that it's pretty close, or maybe we could model it as a 2 or 3 CSTRs in sequence. But for the most part, we're just going to assume that's all well mixed because of all that aeration. Okay, so generally we'll be CSTRs here. Another term we'll use, or you'll hear operators use pretty often, is how strong is your wastewater? You know, it's kind of like how strong is your coffee, right? How much does it hit you when you smell it, right? You could say kind of the same thing for wastewater. That's generally referring to how much organic junk is in there. Bless you. And that we are often going to quantify. That's our S, right? S is substrate. We will often call S as the amount of oxygen demanding substances. So this is generally the amount of carbon bearing molecules in the system in a kind of a loose sense. So this is going to be our biological or biochemical oxygen demand. I should have another slide that mentions that, but just in case. We talked about it last time. We quantify that in a mass of oxygen consumed per amount of stuff transformed, right? The amount of oxygen that bacteria will consume. We'll get more into that after covering the treatment to kind of take a look at what happens downstream once we discharge wastewater. So we'll come back to BOD in more depth. For now, this is milligrams of oxygen demand per liter. And the strength typically varies between something like 50 to 60 milligrams BOD per liter, all the way up to maybe 500. 500 would be really high strength wastewater, whereas if it rains a fair amount here, and we have all these pumping stations for the South Baton Rouge wastewater treatment plant, it tends to get a lot of groundwater infiltration into those pipes, and it tends to dilute it quite a bit. And sometimes they have less than 50 milligrams per liter BOD if it's been pretty rainy. So sometimes their system is quite weak in terms of the strength of the wastewater, which could impact how well your bacteria grow. So there's actually some management that might have to take place there. What kind of reactors are you using? What kind of flow rates are you dealing with? All that. Okay, another term is volatile suspended solids, VSS. This is a way we count our bacteria. So if wastewater strength is S, then this volatile suspended solids is X. Right? This is a mass, milligrams of volatile suspended solids per liter. And essentially what this is you take a filter, you weigh the filter first, you filter a bunch of water, you collect how much mass is left there when you dry it. So that's taking a total suspended solids. This is just you filter, then dry, or evaporate everything. So that'll give you the total suspended solids, but then you take it further and say, okay, well, how much of that can volatilize? Meaning, like, how much of that is not mineral content? How much of it's organic matter that will burn away if you put your oven on the cleaning cycle, right? You roast it at 500 Celsius, which is quite hot. Anything that burns away, we will call volatile. Anything that converts to a gas and runs away. So most of what a bacteria is made of will do just that, right? So this is sort of a crude technique to get a mass of the bacteria that are there, and that's what we use as a way in a lab within maybe an hour or so to do this process to check our bacteria concentration. It's a lot more convenient than doing like a petri dish thing and waiting for 24 hours and getting everything right with all that, doing all the dilutions. This is just a little more cut and dry. You grab the sample, you filter it, and you burn it. You've got to weigh a couple times in between. Okay, so then this is anything that did not volatilize. So we count the difference between the mass remaining and the mass we started with. We're not really going to do any calculations with that. So I'm just going to leave it at that, explain that that's what the deal is. All right, so what can we expect from wastewater treatment? Well, typically the secondary treatment provides us with somewhere between 95, 98%, somewhere in that ballpark, of BOD removal. So if we started with, let's say, 100 BOD, 100 milligrams BOD per liter, primary treatment can take out something like 30 to maybe up to 50%. We'll give it on the low end and say 30% right now. Maybe it's lower than that. So we'll say primary would get us something like 70 milligrams per liter BOD remaining. And then we send it through secondary. So if we took, let's say, 90, 95, 99, 95, 98% of that, we should be somewhere in the ballpark of, someone do that math for me real quick. What's 5% of 70? Should be like 3-ish or something. 3 something? 3.5. Okay, so we'll say 3.5 milligrams BOD per liter left. Now, physically what this means, and we're going to go deeper into this later, but water just sitting here at room temperature in the normal atmosphere can hold something like 8 to 10 milligrams per liter of oxygen. Okay, so if you have equilibrium with the atmosphere and a kind of room temperature-ish condition, you'll be able to dissolve about 8 to 10 milligrams per liter. A little more if it's colder. So this means if you had wastewater at 3.5 milligrams per liter, that would drop our oxygen in here from, let's say, 8 to 4.5. So that means this wastewater that we just treated in this little example is now probably sufficiently protective of fish and wildlife. As long as 4.5 or whatever was remaining is still sufficient for all the fish and everything to grow. So that's kind of the deal, and that's kind of the way to think about this. You will also have a dilution, right? Because your river is not made up of only wastewater. So there will be some aspect there where you will have some dilution of that waste. And you can see if you didn't treat it, you're trying to use up 100 milligrams per liter and you only had 8 milligrams per liter, you're in a bad spot, right? You're going anoxic pretty quick. So that's kind of the scheme of BOD, and that's the ultimately the goal, the primary, the conventional goal, one of the most important goals of wastewater treatment is to take out that oxygen demanding substance, whatever that happens to be. It could be sugars and fats or who knows what. Okay, another kind of tie-in here is most of that's going to be carbon. I kind of mentioned that. When we think about what life is comprised of, there's really 3 elements primarily. There's carbon, nitrogen, and phosphorus. You'll notice that when we fertilize plants, right? The nitrogen and phosphorus are what we are trying to add. Or if we're worried about wastewater discharges, usually the nitrogen and the phosphorus are what we're worried about discharging into the environment that could cause algae blooms and things like that. So a typical ratio for an organism, there's a well-known ratio, the Redfield ratio for oceanography for algae. It's pretty similar I think that one's 108 to 17 to 1 or something. But typically you'll have about 100 parts carbon, 100 times as much carbon as you would have phosphorus. So this would be C to N to P. So if you have 100 carbon, you have something like 10 to 15, well, 5 to 20, I guess, nitrogen and 1 phosphorus. So there's a kind of a generic ratio we can get, and we can find this if we take a look at what cells are comprised of, right? DNA, cell membranes, things like that. There's usually phospholipid membranes, if you recall from that kind of stuff. Some nitrogen and all that. I don't remember what video this is, so we'll see. Forgot to bring it up in the first place. Okay, this is a waste-activated sludge.
At Vistaprint, we can get your brand into your customers' hands, or their heads, or their backs.
For a moment. I don't think we need sound for this one, but. So this is a wastewater treatment plant that's going to be looking in. It's going to go into this chamber on the right. I think is probably might be a primary clarifier, and on the left you can start seeing there's activated sludge system with all those bubbles coming up. So they're going to just walk through it. I think it's just like a 2 or 3 minute, 4 minute video. So they're kind of walking along the treatment train a bit. We have that inflow. On the right here, it's spilling over the top. So this is going to let large stuff settle, whatever can settle into sludge, and it's collecting the water over the top of that surface. That's going to be taken around, and it's going to come under this channel here that we see, and then go straight into this activated sludge. So on the right now, all this here, that's now the activated sludge tank. So you can see the beautiful mixed liquor, suspended solids there, bubbling away. Looks pretty appealing. One thing to note is if it wasn't obvious already, you really don't want to fall in there. But for more reasons than you might imagine. So not just all the grossness and the gross factor and the dirtiness, not just because it's probably like 15, 20 feet deep, but also because you'll have no buoyancy. Because most of what you're seeing there is all those air bubbles. When you try to displace water and air, you can float on water pretty well. You can't float so well or swim so well when it's there's so much air occupying all that volume. This is also the reason that if you ever are on a sinking ship, you actually should swim away from it if it's going down, because there's going to be a lot of bubbles. Could sink you too along with it. So there are protocols to anybody working in a facility like this to know where the ladder is, because there should be a ladder every so often as a way to escape, because you're going to have to kind of walk your way without being able to see anything to that in order to escape. So it's actually quite dangerous. So that's all the railing. So at this point, with all that beautiful, wonderful thought in mind, here's probably the secondary clarifier. It's what we see on top is just sort of some scum that floats up at the top. That's generally going to happen so long as most of the bacteria aren't forming like that scum stuff. We're okay. If sometimes we have a situation where a whole bunch form like floaty stuff, and it becomes a big problem. Here on the left, I think that may be a chlorine contactor if they're doing disinfection. It might just be another step of the clarifying. But by the end of it, you see what's coming out is actually looks pretty reasonable, more like what you might find in a river, right? So it's actually kind of just that process seems to be doing pretty well there. Just gonna fast forward a little bit, better picture here. Yeah, it almost looks like that's river water, right? So the treatment can do pretty well, and if you disinfect it and you're monitoring for whatever else you need, it's not too bad. I have an example here, just a kind of a note for myself. I had a chance to tour a tissue recycling plant while I was working as a permit writer. And so this is like, taking cardboard, taking any paper materials, paper products kind of stuff. And what they would do is they'd send it into some sort of grinder, shred it up a bit, send that into a big vat of chemicals to dissolve everything and to essentially get down to the fibrous tissues. It would then go through several more processes, and then you'd start collecting all the tissues together and all that. Ultimately, they'd end up able to create the type of paper towels that we use in the bathrooms and stuff here. They'd be creating products like that. And on the flip side, they had a wastewater stream. And that wastewater had primarily this like carbonaceous tissue stuff, right? If you imagine like taking tree material, you grind all that up. It's a lot of carbon there. It's not quite the same as like taking a piece of fruit, right? You can maybe even visually or like tangibly think that, okay, one of those is edible, the other is going to taste like cardboard, right? You kind of even almost have a feel for what that means, right? So it's primarily carbon stuff. So they didn't have very much nutrients in terms of nitrogen or phosphorus. But they needed to remove this carbon. So in this case, it's kind of an industrial example. They had to add they would literally pour fertilizer into their bioreactor with a certain quantity to feed the microbes the nutrients that they needed in order to grow. So their wastewater would have discharged a lot of BOD without much nitrogen or phosphorus. They had to handle that, so they actually had to add nitrogen and phosphorus to let the bacteria grow in order to do that function for them. And that was still the more cost-effective way. Then you probably also have a slight risk of adding nitrogen and phosphorus to your effluent, so they obviously wouldn't want to add too much there. Okay, so that kind of stuff is relevant in general. I wanted to give you a picture of what it takes, what proper nutrition for microbes, I guess. In general, municipal wastewater has plenty of nitrogen and phosphorus. In fact, oftentimes we run into the problem where there's too much, and we really don't want that going out to the environment. So, but there are cases you'd be surprised how many different industries have random issues for their wastewater treatment that perhaps you can use an adapted version of what we're talking about here, which is kind of the general municipal wastewater treatment setup. Okay, so how do we know about the growth of the microbes? So we have an equation that's pretty well known in the wastewater field called the Monod equation. And this is an equation that describes the rate constant for our bacterial growth. Okay, so we're going to do a bit of mass balance on how fast bacteria are growing, right? We've done mass balances on how fast flocks are growing in flocculation, except we did that in reverse, right? We did how fast are the particles being removed. And if you remember from that rate constant depended on several other things, right? So sometimes we get this rate constant that depends on a few things. In our situation here, you can imagine bacteria are going to grow faster if they have a lot of food, right? So there's going to be some issue of how fast can they grow given how much food they have, maybe even how much mass of bacteria we have. Okay, so that's kind of what this equation is going to describe. And this mu here, I'm following the book's terminologies here, this is basically our K, or rate constant, for our bacterial growth. So if we think for a moment, just simply how would we describe bacteria growing? We would say, well, the change in bacteria over time, dx dt, is going to be related in some way to how much bacteria we have in there plus some rate constant, right? So it'd probably look like that. Dx dt equals mu x. So that's what mu is here serving for us is kind of a rate constant. I just want to let you know that's kind of where we're at with this equation and what it's going to do for us. We'll see it in a few spots, but that's where we're headed. Okay, so this mu equals mu max, that's mu m, times the food, the substrate there, divided by some constant Ks plus the amount of food. Okay, so we've got this equation, and it relates to how much food we have there for the microbes. So the mu max here, this guy, if we look, that's the maximum specific growth rate coefficient. This is per time units. This is what's going to give our overall rate constant per time units, right? So mu should be in 1 over time units. So the mu max then, that's the case where if we gave bacteria an infinite amount of food and the ideal conditions and all that, this is how fast they're growing, or that's their maximum specific rate constant, growth rate coefficient. Okay, so if we have mu as the generic specific for this particular condition, mu max is the maximum specific growth rate coefficient. All right, so S we've talked about. This is the food. That's the stuff we're trying to remove in this whole deal, right? That's the purpose of it. So then the only thing remaining is Ks. Well, Ks is called the half velocity constant. It's also a measure of food, and it turns out that this is the amount of food where the bacteria are halfway happy, halfway to their maximum growth rate. So I have a graph on the next page. We'll take a look at how that ends up looking like. It's something we can measure. We can say, well, at what point do the bacteria no longer need any more food to grow? Maybe that's 100 milligrams per liter of BOD. Who knows? Whatever that value is. Half of that, if we find, it's not just half of that food value, it's where, like, we found that the bacteria are growing at some rate. Let's half that rate constant, and what's the food looking like at that half the rate constant value? So if we had, if we plotted mu and we had some max value, mu max, and we are giving bacteria more and more food, we're going to start with 0 as a rate constant, right? It's no food, no growth. And it will grow, this will increase pretty rapidly, and then eventually kind of plateau and meet mu max. So what we're saying is at half of the rate constant here, this would be mu 1/2, I guess, at some point, that corresponds to some amount of food. And so we can look down here and say, okay, that's Ks. So it kind of feels a little bit arbitrary, but there's a physical meaning to it. That's just kind of what it is. So if we take a look at our equation, the rate constant is going to be the amount of food we have times the fastest they could possibly grow, divided by the amount of food they have plus the amount of food where they're halfway to their max. So it's some ratio. If we had an infinite amount of food, then we'd have basically infinity divided by infinity, and those would kind of cancel, and we'd just be left with mu is equal to mu max. Right? If we had a very small amount of food, then it would be like, okay, we have some ratio here that is a mu is equal to mu max divided by a big number. Because if S is very small, it's kind of like 1 over Ks. Or even worse, it's like 0.000 something, maybe, divided by Ks. That turns into a big number on the denominator, essentially. So if we take a look at it, it just is going to change our current mu based on how much food we have. It's just a fancy way to do that. So that's kind of what it means physically, or how we could measure it. For the most part, we'll be given some value. We'll be given a Ks. Maybe we solve for Ks sometime, but we'll be given most of these parameters to say, okay, here's what we're working with. So I just wanted to let you know that. And by the way, this is a capital KS. We're going to deal with a few different K parameters, constants and such. This is capital K subscript S. Okay, so when we take a look at how the bacteria grow in practice, there's more than just the growth that we end up needing to take care of. And here's kind of an example of what happens if we take a beaker of broth, or solution of wastewater of something, grow bacteria in it, give it oxygen, give it the food at the start, and we have some amount of bacteria to begin with. Well, if we just let that grow for some time, what we'll end up with is a scenario where maybe it takes a couple of hours before the bacteria kind of wake up and start eating and reproducing and doing all that. So there's often a little lag phase. That's more relevant in a lab scenario. If you have wastewater ongoing, then would not be so relevant. In wastewater, we want to keep some happy bacteria that are already growing. So we want to make sure we're not encountering that lag phase. That's one reason to recycle the wastewater. So on a log scale, this is the log of the number of bacteria. We're going to hit this acceleration phase after a few hours where we are growing exponentially. So it looks linear on this plot, but that's because we're on a log plot. So that is exponential growth, and it's, excuse me, that's what we call the growth phase. At some point, we're running low on food, and it's really crowded, and so the bacteria end up starting to compete with one another, and the whole time there have been some bacteria just dying. That just kind of happens. They don't live super long time. So this whole time there have been microbes dying, but they're just growing so fast that it's way outpacing the death. At some point, we end up with a stationary phase. Well, we'll have a decline phase where it's no longer exponentially growing. There's some curve there. We'll hit that stationary phase, and what we could describe is we'll say R of growth, the rate of growth, is equal to the R of decay. Whereas over here, we're just basically seeing R of growth. It's much bigger than the rate of decay. And towards the end, we're mostly just seeing, after all the food has been consumed, the only thing left is for some of them to start dying off. They'll probably cannibalize each other's corpses and, continue to grow a little bit, but they're mostly just dying off. So that's kind of the scheme of bacterial growth in a lab setting, like if you were to take a container and do that. So in practice, all of these are, both of these rates are still at play. So in a wastewater treatment scenario, we do want to understand something about both the growth and the decay there as we go. Related to this, and we'll come back to this in a little bit, is the thing that we really care about for our treatment is at what rate are we consuming the waste. So there's a rate of substrate utilization that we can derive from this growth rate. So not pictured here is rate of substrate, not subrate, substrate utilization. So you might imagine the speed at which they're growing, the amount we have growing, will impact at what rate are we consuming the S, the food, right? So we'll come back to that. All right, so the growth rate. I already kind of glanced at, showed us this. We took a quick look at this earlier. dx dt would be a way we can describe the change in bacterial concentration over time. And we could describe that as mu x, mu being our specific growth rate coefficient, x being the amount we have. So this is very much like any of the reactions we've seen, except this is a growth reaction, so it's going to play out a little different than a decay. We also have a rate of growth. I mean, we could call that a rate of growth. So Rg is equal to mu x, and if we substitute the mu, that Monod equation here, that's this piece, right? So all I've done here is just expanded. Instead of putting mu, I put in that entire equation we had for mu. So now we have a definition of the rate of growth as x times mu max times the substrate, all that divided by Ks plus S. Okay, so that gives us a rate of growth for our system. Now, when we want to compare this and find this rate of substrate utilization, what we can do is we can recognize that there's some sort of factor. You feed bacteria, X grams of food, well, you'll get Y grams of bacteria mass, right? You could consider this with all sorts of things that are actively growing, right? If you have fish in an aquarium and you feed them a lot, they'll grow bigger faster than if you don't feed them very often, right? And if you do want happy, healthy fish in your aquarium, you generally don't want to feed them too much, presumably unless you want to try to make them bigger. But for the same reasons, all these nutrient cycling things, you don't want to give them a whole bunch of biological oxygen demand by overfeeding them. It kind of creates a wastewater scenario in the aquarium, right? So there's some aspects there. We can imagine you can get some yield. So we'll have a yield coefficient for the amount of mass of bacteria we grow per food that they're consuming. So that's what this lowercase k with no subscript. Note that this is not talking about the Ks, right? So scratch that from your memory for a moment and say lowercase k is our, this is going to be a way we can designate the amount of substrate utilization. So K will be our maximum specific substrate utilization coefficient. That's going to be equal to mu max divided by Y, which is a yield coefficient. So all that to say, we're going to use the substrate according to how fast the bacteria are consuming it in relation to how much mass of bacteria we get. So right here is mass of bacteria per mass of food, right? So if we take our maximum specific growth rate coefficient, divide that by this yield coefficient, that's our maximum specific substrate utilization coefficient. So in that gives us a value of mass of food per mass of bacteria per time. And usually we'll use days for that time. Okay, so that's kind of just feels like a lot of jargon, a lot of just labels and units and stuff. But what we just want to recognize is here's, a study up on, okay, what parameter is this? Which one is which, right? I haven't posted yet the next formula sheet, but I will soon. So you'll have that and you can study, okay, what is this lowercase k? What is the uppercase ks? Be able to work with those so you understand where they're piecing together. And with that, we can relate our rate of substrate utilization to our growth rate. So the amount of substrate we're using is going to be proportional to the speed at which we're growing. But we're going to divide that by that yield coefficient. So it's like if we are growing 200 kilograms of bacteria every day, and each kilogram takes like 10 kilograms of substrate, then that 200 kilograms of bacteria correlates to 2,000 kilograms of food, right? That's the type of relation we're doing here. It's like, okay, how much do we yield given this amount of food? And we'll do that comparison. So, and that's going to be a negative correlation, right? So the rate at which we're growing, so the substrate utilization, that's a negative, right? We're consuming the substrate. Growth is positive, so that's why we have the negative there, and then it's dividing by that yield. Okay, so we've covered now the growth, and that relates to our substrate. Well, what about the death? Because we're going to have to incorporate a rate for death and decay of our substrate or of our bacteria as well. Okay, so here we have yet another rate constant k. So R of death is going to be simply negative kd times x. Right, this one will be pretty simple, straightforward. All of these are going to be first order, you'll notice. So kd would be the death and decay rate constant in per time. So that one's pretty straightforward. And again, we'll have some constant for this. I'm sure there are factors that it depends on, but typically those will just be known, published, or something that an estimate would do fine for. So net growth rate then, R prime of g. So we want to know on the whole, how much are we growing? So this is going to combine the death and the growth. What we can do is relate, instead of using the growth, we could actually input the substrate utilization rate here. And then we're in a situation where we were doing a mass balance on how much bacteria we're growing, but what we end up learning is how much substrate we're using. Okay, so there's that relationship between the two actually becomes pretty helpful. So let's walk through this. The net growth rate, net change in x, R prime of g, can be described as R of g plus R of d. And remember from the last slide, we said that our substrate utilization rate was equal to negative Rg over y. We can solve this for Rg, and we get this, right? We get R of g is equal to negative R substrate utilization rate times y. So we can then substitute that R in here, and we get that. So if we do R prime of g, and we want to, in terms of substrate utilization, this is going to be R prime of g is equal to negative y times R of substrate utilization plus a negative k dx. Make sense? Okay, so this is what we've described so far with the growth and death kinetics, all this. This is for a scenario of just simply bacteria growing. We're going to put it in the context of this, clarifier, waste-activated sludge, recycle system. We're going to put it back in that context, not this lecture. We're going to come back to that. First, we're going to deal with, well, what's it like if we just look at the growth kinetics in a generic system, without the fancy recycle, without all this engineering control. So this example here, example 6.9, page 322, gives us, from our book here, gives us a bunch of these parameters. So if you take a look here, we've got K, we've got mu max, we've got Ks, we've got Y, and Kd. So all of these parameters, this table is giving us typical units for those parameters. So we could use this as a lookup, say, okay, well, we've not given a mu max, so we're going to estimate it's somewhere between 1 and 10. Maybe use this default value here. Let's say 3 per day. So it gives us the units, gives us some default values we could use. And then this problem says we've got a shallow pond here depicted, and it says well mixed due to wind and steady flow through of a small creek. I think I have the problem text on the next slide, but essentially what it's going to ask is what is the final concentration of X and the final concentration of S. So how much bacteria grow in this scenario and how much substrate was consumed in this system. Okay, so what we can do with this problem, and I'm going to give you a little bit of time to take a look at and to think about how you might approach it. This is a complicated one, and we're going to have to do a couple different mass balances for it. But we'll work through it together. I'm going to give you some time to think about it first. So the problem says if the microbes in this pond consume the inflowing biodegradable organic matter according to typical kinetics, determine A, the substrate leaving the pond, they're going to call it BOD5. B, the biodegradable organic matter removal efficiency of the pond. So if A is asking for S, B is asking then for what's the removal efficiency. This is going to be pretty generic, right? What we ended with, S, let's see, S naught minus S. What we started with minus what we ended with, divided by what we started with, times 100%. That would be a pretty generic way to get the efficiency. And C, the concentration of volatile suspended solids leaving the pond. That's looking for X. Flipping back for just a moment, we see that it says it's well mixed based on wind and stuff. So X here is equal to what's leaving, and S here is equal to what's leaving for S, right? So we can make that relationship. And this is essentially then a CSTR. We're given some parameters, but the deal is we're given both bacteria that's changing and substrate that's changing. So we're going to do some mass balance magic to simplify and find out what's really going on. Okay, so the solution, I'm going to go ahead and start there. We'll let you take a look at that for a couple minutes, and then I'll work with you on it. So it starts by saying the pond can be modeled by a steady state CSTR because it's well mixed, and we'll assume the flow has been steady for some time. Part A says, first set up the mass balance on the microbial mass using the pond as a control volume and variables in figure 6.14. So given that, we can write for a mass balance, we're doing a mass balance on microbial mass. So 0 equals Qx naught minus Qxf plus V times the net growth rate equation there. So one of the tricky parts about this problem is what you will find is in order to solve for S, we do a mass balance on X. And to find X, we do a mass balance on S. Because they relate to one another. It's a little bit counterintuitive, but that's where they're starting here, so I wanted to go ahead and give you that tip. So I think I have, okay, all of that a little bit smaller here. I'm going to write down what they started with right here. I'll write that up for you and give you a few minutes to look over this. Does that also have X? All right. I might flip back again in a moment. So R prime of G, actually, Yeah, I'll put in the kind of the standard values in a second. Let me grab those. You can get pretty far with just the just using the labels, right? So just using the symbols for to start. All right, everybody have a chance to give that some thought? Do you feel like you were getting there? Maybe? Okay, so I realized that when I was putting up the equation for R prime of G, sometimes we will need that to put it in terms of substrate utilization rate, and that's helpful. But I also recognize that even just mu x here might be the simpler way. This is the RG, right, minus RD to get R prime of G. Sometimes we'll need to substitute to use that Y factor. We might come back to that, but for the moment in the first step of the problem, I think we actually need the direct substrate values here we can get from treating it as just the growth. So the growth is mu x in that volume, and so we've got what I've done here with the mass balance. I said 0 equals qx naught minus qx plus in that volume the net growth rate. So what's coming in is actually 0 because x naught is 0. We're assuming there's some x in the pond for them to actually grow on, but we're starting with, the influent has 0, so that'll be 0 equals 0 minus qx, and then I'm going to go ahead and expand out this to that volume times mu times x, which is the growth rate equation, minus the death rate equation in that volume as well. So with that, we can go on and say 0 equals minus qx minus v times x times mu max s over s plus ks. All that minus v times kdx. So a couple things to note here is we can start simplifying. And when we start doing that, we look at common terms, something really nice happens, all because we had a 0 here. If we didn't have a 0 there, we couldn't divide everything by x and completely get rid of x. So what we're going to continue on from here is for the no recycle case. Because we're not recycling any bacteria, the mass balance works out pretty convenient. So if we do problems, we'll notice recycling the bacteria makes a big impact on how the equation works out and on the outcomes of the system. It turns out that when we do recycle, we have some other shortcuts to simplify our math and what we need to do with our reactors. But in this particular case, without the recycle, it's convenient when we're trying to approach it from a raw mass balance perspective. So we're going to have equations that equip us to deal with the recycle. It will feel a little bit less mass balancey in terms of implementing those equations. This one is pretty hardcore mass balance. Like, okay, we've got these things. We have to figure out, okay, there's no x in the influent, and how do we do this one and then the other one and all that. So there's going to be kind of two different systems we're working with. One is recycle, no recycle, and the other is with recycle. So we're going to come back to with recycle next time, but I'm going to continue here to show that as we go from here, we can divide everything by q and everything by x and simplify quite a bit, right? So if we divided everything by qx, this gives us minus 1 there, and minus v over q is theta times, we got rid of the x, so this is mu max s over ks plus s, minus, again, we've got a theta here, kd. All right, so at that point, we have a much simpler system. X is unknown, but now it's gone. S is still unknown, and that's what we're needing to solve for. So at this point, we just solve for S, which was kind of the goal in the first place, right? So if we simply add the term with the S's, from here it's going to be algebra, and I'm going to have my previous notes where the proper solution derives. So if I go astray, I can remind myself here. But basically, we just need to solve for S here, and it's not the most convenient thing, but we'll work it out. So theta mu max times S over ks plus S would be equal to -1 minus theta kd. So just need to get the s's by themselves, so I'm going to go ahead and multiply both sides by that ks plus s. And from here, we'll say that's, I'm just going to multiply them out. There might be a simpler way to do this, but I'm not sure at the moment. So that'll be ks minus theta kd ks minus s minus this theta kd s. All right, so at that point, we're going to add both the s terms to the other side so we can factor them all out. So I'm going to give ourselves a little bit of space here. To work with. Sorry for the mess here. We're going to say theta mu max times s plus s plus theta kds is equal to this negative negative ks minus theta kds, theta kd ks. Okay, so we'll get the s terms, we'll factor out the s, we'll be left with theta mu max plus 1 plus theta kd equal to negative ks minus theta kd ks. All right, so finally we can get s equals minus ks minus theta kd ks all over this theta mu max plus 1 plus theta kd. Okay, and I think we can simplify that a little bit more. If we pull out that ks from the top, and I think I flipped a negative somewhere.
Is it when you did the, like when you plugged in mu, you have a plus b mu x, but then the next time you have minus. And I was wondering why.
Yeah, that should be it. Thank you. That was not supposed to be there. So I think that would change that. Okay. I think that fixed it. I do know from my prior notes what it looked like was ks times 1 plus theta kd ks. All that over what I had on the bottom was looking correct. Okay, so kind of involved. I made some algebra mistakes. You know, we ultimately get to this value, this mass balance. We did a mass balance on x to solve for s, is effectively what we did. Kind of backwards. This is for a case where we have zero influent bacteria mass and no recycle. So given that all those things are true, then this equation for S will hold up. So I don't remember, I think maybe I gave it to you on the equation sheet, but you should definitely make sure you know that's a very specific derivation for a specific condition. The pond example, right? There's no recycle. Maybe we'd work through a problem like that, but that's this particular example. So when well, that's not the final answer here. When we put in all the numbers, S should come out to be a value of 6.9 milligrams BOD per liter. That's the actual answer. So then we can calculate the efficiency and then finish part C. So we'll pick up there on Tuesday next week. All right, see you then.